Assalamu alaikum guys, welcome to my channel. This is Faris Al-Shbul talking. I am an internship doctor at King Abdullah University Hospital, and this is the first video of a series that will talk about the field of microbiology based on Burton's microbiology textbook, the 10th edition, made by Engel Kirk. The series will be primarily in English, however there might be some Arabic words here and there. In this video, I will be talking about the first chapter of the material, which is just a simple introduction to microbiology. And the next part, I will finish the chapter by talking about the branches of microbiology and some history about the microbiology pioneers. Okay, let's begin, shall we? Okay, so this is chapter 1, Microbiology the Science. Here are some terminology. Microbiology comes from Greek. Micros means small, bios means life, logos means the study of. So this is the study of small life forms, the study of microorganisms or microbes. So what are microorganisms? Microorganisms are the organisms that are too small to be seen with the naked eye. We need a magnifying lens or a microscope in order to see them. So what is microscope? Microscope is an instrument used to see small objects that can't be seen with the naked eye. So what is microscopic? When we say something is microscopic, what does we mean? What do we mean? Microscopic means the object that the object cannot be seen with the naked eye, so it is too small. Indigenous microflora. These are the collection of microorganisms that live on and in the human body. These are normally found and they don't cause any problem. In fact, they are helpful. Okay, the pathogen or a germ is the microorganisms that is bad, that causes disease. So most microorganisms don't actually cause disease, while some of them, only 3%, are pathogens and cause disease. These diseases are two types. We have the infection and the intoxication. The infection is the disease caused by the pathogen itself. The intoxication is the disease caused by the toxins produced by the pathogen. We have the opportunistic microorganism. These are microorganisms. Opportunistic in Arabic means intihazi. So these are microorganisms that are non-pathogenic under normal circumstances, but when the opportunity arises, they become pathogenic and cause disease. Such opportunity is like the immune system. Whenever it is weak, they become pathogenic and cause disease. Okay, again, the difference between the infection and intoxication. Infection is the disease caused by the pathogen itself. The pathogen enters the host, colonizes it, and causes disease. While the intoxication, التسمم, when a toxin produced by the pathogen outside the host enters and causes disease. So the toxin is the cause of intoxication, while the pathogen itself is the cause of infection. Keep in mind that outside the host means in vitro, and in vivo means inside the host. Okay, now into classification of microorganisms. We classify microorganisms in multiple ways. One way is to classify them according to pathogenicity. So we have the pathogens that comprise only 3% of all microbes, and we have the non-pathogens that comprise 97% of them. Some non-pathogens are beneficial, like the indigenous microflora. Now we have some factors that affect the risk of infection by a pathogen. So let's assume some bacteria entered our body, some pathogenic bacteria entered our body, and what is the likelihood, what is the probability of this bacteria of causing infection? Well, we have three factors affecting that. We have the infectious dose, which means how many pathogens entered the body. The higher the number, the higher the pathogens, the more likelihood of infection. We have the virulence factors, which are materials produced by the pathogen to help itself causing disease. So, for example, these bacteria might have pili. These pili are hair-like projections that help it attach to surfaces. Also, they might have flagella. This flagella help the bacteria in locomotion, in movement, so they can evade the immune cells and cause disease. The third factor is host factors. This is mostly the immune system. So if the immune system is good, the risk of infection is low. If it is weak, the risk of infection is high. Okay, another way of classifying microorganisms is according to cellular structures. So when we say cell, a cell is the thing that is comprised of cytoplasm and surrounded by plasma membrane. So anything that has cytoplasm surrounded by plasma membrane can be considered cell. So some microorganisms are cells, okay, but others are not. These non-cellular, we call them acellular microorganisms or infectious particles, are like the viruses, the viroids, and the prions, while cellular are either prokaryotic or eukaryotic. Okay, so firstly, let's discuss the acellular. The acellular microorganisms, for example, the viruses. Okay, so the virus we all know or hear about them. These are very tiny. In fact, all the acellular microorganisms are very tiny, very small. So viruses are genetic material, DNA or RNA, that are coated with a protein coat. So what they do, when they infect any cell, for example, human cells, they inject their genetic material in that cell and use that cell for their production. So these acellular they depend upon other cells for their reproduction. Okay, another name for viruses is virions. Now the viroids, anything that ends with oid, oid, means something like. So viroids is something like a virus. So it is a virus, but without the protein coat. The prions are misfolded proteins that can cause disease. One example for diseases caused by prions is the mad cow disease. Now moving on to cellular microorganisms, these are subdivided into prokaryotic and eukaryotic. Prokaryotic means before the cell, before the nucleus, and eukaryotic means a true nucleus. So the prokaryotic have a primitive nucleus. Their nucleus does not have nuclear membrane, while eukaryotic have a true nucleus and have a nuclear membrane. Other difference between prokaryotic and eukaryotic, eukaryotic have membrane-bound organelles. For example, they have mitochondria, they have endoplasmic reticulum, they have Golgi apparatus, etc. While prokaryotic microorganisms, they don't have organelles. Most of them don't have organelles, for the exception of cyanobacteria. Okay, so the microorganisms, as I just said, are acellular, viroids, prions, viruses, or cellular, which are subdivided into prokaryotes and eukaryotes. So the prokaryotes are primarily the bacteria, and eukaryotes are either algae, protozoa, and fungi. Algae is طحالب, and protozoa طفيليات, and fungi the فطريات. So the prokaryotes, I said it's primarily the bacteria. Now, some bacteria, okay, some advanced bacteria can produce oxygen, and these are called cyanobacteria. This is the only prokaryote that have some sort of organelle, okay, that help it produce oxygen. While other prokaryotes, most prokaryotes never have any organelle. Okay, now there is a type of microorganism that is considered like a brother to bacteria, and it's called archaea. Now, archaea and bacteria once were one thing. They were one family, one kingdom, one domain, um, but now we separated them for some reasons. Okay, these are some examples of viruses. Viruses can have multiple shapes, and these are some shapes of viruses. Okay, the bacteriophage has a complex shape, and it infects a bacteria. Now, this is a diagram of bacteria that are resistant to antibiotics. Escherichia coli in particular is the microflora that is the main microflora of the GI tract. Now, bacteria and archaea look alike. They look the same. They are both prokaryotes, but they differ in three main things. Well, the bacteria have peptidoglycan cell layer. The archaea don't have that. The bacteria can be pathogenic. Some of the bacteria cause diseases, while archaea don't cause disease. Bacteria live under normal circumstances, normal living conditions. Archaea can sustain extreme living conditions. For example, archaea can sustain high levels, very high concentrations of salts, like those in the Dead Sea, and it can sustain high temperatures succeeding the boiling point of water and below the freezing point of water. Now, the archaea and cyanobacteria are considered the first microorganisms on Earth. This is the algae. Algae can take multiple construction types or multiple shapes. We have the unicellular algae like this one and this one. We have the colonial algae, this one. We have the filamentous algae, this one, and we have the multicellular macrocytic algae. This is not microorganism. We can see it clearly by our eyes, while these ones are microscopic. Sorry. Okay. Now, what's the difference between algae and plants? First, they are both eukaryotes and both photosynthetic. They produce oxygen. But they differ in three main things. First, plants have tissue differentiation. They have roots, trunks, and leaves. While algae, all of the cells have the same structure and the same function. They don't have tissue differentiation, even with this multicellular algae. All of the cells have the same structure and the same differentiation. Algae can be unicellular or multicellular, plants only multicellular, and algae live underwater, plants live on land. That's very obvious. Now, the protozoa, these are parasites. They depend upon other organisms for their reproduction and their nutrients. Entamoeba histolytica, Giardia intestinalis, they cause chronic diarrhea. Trypanosoma canviense causes African sleeping sickness. Plasmodium vivax causes malaria. These are the fungi, the shapes of fungi, the types of fungi. We have the yeast and the mold. Yeast is الخميرة and mold is عفن. Yeast is unicellular fungi, while mold is multicellular. They have the typical hyphae structure. And we have these are the microscopic fungi, and this is the mushroom, is the macroscopic fungi that we all know. This is the yeast, this is the mold, and this is the mushroom. Now, with the most important question, why do we care about microbiology and why we study microbiology? First, microbes are ubiquitous, meaning microbes are everywhere. In our body, we have 10 trillion cells, okay, in our body, but we have 100 trillion microbes in there. So for every one cell in our body, there is 10 microbes. Also, in 1 gram of soil, we have 1 billion of microbes there. The other reason, some microbes are helpful. The third reason, some microbes are bad, pathogenic. They cause disease, and we want to understand disease. And the fourth reason, some microbes help us produce things. Okay, the biotechnology is the technological use of microbes for our help. Okay, so how some microbes are helpful? Well, we have the microflora, the indigenous microflora. They help in digestion, in vitamin production. They help, for example, some vitamins, vitamin K and vitamin B are produced by the microflora. For example, the Escherichia coli. And also microflora reduce or prevent pathogenic growth. How do they do that? Well, they occupy the space and take the nutrients that this will prevent the pathogens when they enter from multiplying. Well, the pathogen when it enter and they don't find any space to multiply and any food to feed on, they can't sustain life there. Okay, some microbes also produce oxygen. For example, the cyanobacteria and algae. Some microbes do the most important function of nitrogen fixation. So what is nitrogen fixation? Nitrogen fixation is the process of converting nitrogen gas from air into ammonia in soil. So the air, we all know, is composed of 21% oxygen and 78% nitrogen. So this nitrogen gas, okay, is very important for all living creatures, because nitrogen is a component of genetic material, DNA, RNA. Also, it is a component of protein. And how we get nitrogen atoms in those structures? Well, we get that from ammonia. And from where we get ammonia? We get ammonia from air, from nitrogen gas. And the only living creature that can convert nitrogen gas into ammonia in soil is the bacteria and its brother archaea. So nitrogen fixation. Okay, nitrogen fixation helps in soil fertilization. Another help from microbes is the decomposition. Well, some microbes are decomposers. For example, the fungi and some bacteria are decomposers. They decompose waste material, okay, into nitrate, ammonia, phosphate, CO2, and water. This help us get rid of waste, organic waste, and also it helps in soil fertilization. Such decomposers are called saprophytes or saprotrophs. Okay, now for the biotechnology. It is a technological use of biological systems, for example, the microbes, to our help. So some microbes produce food and beverages. For example, Lactobacillus acidophilus is a type of bacteria that can convert the sugar in the milk into acid in yogurt, lactic acid. Also, we have the yeast fungi that can convert grapes, sugar in grapes, into ethanol in alcohol. So they help in alcohol production. Also, some microbes produce deadly substances, okay, to kill other microbes. So, for example, some type of fungi can produce penicillin to kill bacteria. So what we do, take these fungi and grow them and purify the substance they produce, the penicillin, and other antibiotics. So also other bacteria can produce other antibiotics. So microbes can help us in antibiotic production. Okay, this is the last slide of this part. The role of saprophytes, we just will summarize what we just said. Saprophytes are the decomposers They help us in organic waste degradation, wastewater purification, in soil fertilization, in the production of nitrate, phosphate, CO2, and water, and some of them are nitrogen fixators. Okay, so this is a plant, and this is its roots. You can notice these bulges, these are collections of bacteria. So this bacteria and this plant have a symbiotic relationship. The plant provide the bacteria with the nutrients, the sugar, and etc. And the bacteria provide the plant with the nitrogen. So these are the nitrogen fixating bacteria. Thanks for watching and listening, and don't forget to like and subscribe. And the next part will talk about the branches of microbiology and the history of microbiology, finishing the first chapter. See you soon.